Hello, good morning. Welcome back to my channel. Today let us see about what is gene. Little information we know to increase our information regarding this one. If we see plants, animals, insects and human being, every living creature in this world is built up by the group of cell tissue, and the tissue is made up by the group of cells, and every cell in middle it has a nucleus, and the nucleus contains the genetical information that is called as chromosomes. If we unravel the chromosomes, or otherwise if we open up the chromosomes in simple language, it is like a thread like structure which contains the chemical basis of information. This is called as DNA, deoxyribonucleic acid. So if we see this DNA in a large picture, it has four chemical bonds in which form the information of the human being or any living creature is stored. The first one is the adenine. Adenine tie up with the thymine bond, and also the guanine. The guanine has a chemical bond with the cytosine. These four chemical bonds present in DNA, but whereas in RNA, in place of cytosine and uracil is present in RNA. In site of C, uracil is present, and these chemical bonds, these four chemical bonds are called as nucleotides, and these nucleotides builds up a chemical information or blocks in DNA. That's how the information is passing from parents to you, and this total it is called as a gene. This total block is called as gene. So now in gene, two things are there. What are they? Promoter and transcription regions. Two areas are there. So one is promoter area and one more is transcription area. The promoter area is the starting place of the DNA is called as the promoter area, and followed by the transcriptional area. What is the location of the gene? Nothing but wherever the gene is located, that depending upon that location of the gene, this is called as locus. If we speak about this gene chemically, it has different chemical bonds. Those are, as we have seen already, adenine, thymine, guanine and cytosine. So this information gives the information to the cells to produce the desired proteins. For example, if we speak about the eye colors are brown, black and blue colors. So how this information is passing? By the help of the genes. So if we speak again in detail about this gene, it has two parts. One is axon and one is intron. This, if we take the gene strip, axon and introns are present. So the axons are having the genetical information, means code is there, whereas in introns there is no code is there. So there is no purpose of introns. Still now the researchers told that there is no purpose of introns have found, but axons are having a genetical information like A, U, G, R, T, like that. Okay, so what is happening in this one? Before preparing the protein, splicing is nothing but the separation of the introns from the axons takes place. Okay, so after this one, this is called as intron removed. So next step is translation. So in this, all the axons together form a long strip. Together these axons form a long strip. This process is called as transcription or mRNA. What is this mRNA? Messenger mRNA. How this messenger mRNA formed, we will discuss in the next video. So after that one, translation. So from this one, the desired proteins will be formed. So, and one of these genes character is it gives the genetical information to their offsprings and from there to their children. So like this, the genetical information is passed from one generation to the other generations, and it continues for the generations to the generations. Now let us see protein synthesis or making protein. In making protein, there are two processes are there. One is transcription, another is translation. What is this transcription? Reading out a single strand gene and making mRNA, messenger RNA. Translation is like reading out the mRNA and with that help making the protein. So let us see in detail about these two process step by step to make you understand clearly. So now every cell has nucleus, and in nucleus genetical material is present, that is DNA. So in DNA there are like strips of genetical information is there. These are all called genes. So to make, so these genes having a specific sequence, as we have known that adenine, guanine, thymine, uracil, these are all called as amino acids or amino acid sequences, as we have. Okay, so to read this one we need ribosomes which are outside of the nucleus in the cytoplasm. So let us see in detail about, look, so if you see this DNA, it is so big and cannot leave the nucleus to ribosome. So to use a gene, to we have to make a copy of it. That means we have to make a single gene copy to make the protein, and it can leave from the nucleus to the ribosome, whereas DNA cannot leave. So we are speaking about the mRNA. Messenger RNA is the copy of gene we are speaking about. The RNA, mRNA just looks like a DNA, but some important differences are there between DNA and mRNA. It is much smaller than DNA because it is a single copy, and it also a, it has single strand rather than double strand in RNA. And instead of thymine, it contains uracil in place of thymine. Okay, these are all some differences between the mRNA and DNA. So now we have the basic concept of the information. Now let us see into the transcription. What it is actually transcription? If we see the DNA, a single strand, it looks like this one, and if you uncoil this one in a simple diagrammatic, it looks like this way, the double stranded, which contains the chemical compositions like guanine, which pairs with the cytosine, and adenine, which pairs with the thymine, and so on. So this is a simple diagrammatic representation of DNA which presents in the nucleus of the cells. So in transcription, what is happening in transcription? If we think, okay, this is the single piece of the information that we wanted to copy. So what happens to make it copy? A ribosome, an enzyme, RNA polymerase comes and attach to this DNA. A DNA polymerase comes and attached to this DNA. And as it is moving in DNA, the strips will get separated in DNA. And also, this enzyme, RNA polymerase, starts reading this code. As it is moving forward, the DNA strands will get separate and the information in this is read by this polymerase. See, here in this DNA, if we see the cytosine paired with the guanine and then guanine paired with the cytosine, but whereas in mRNA it is reverse because thymine is not there. In place of adenine, adenine will pair with the uracil and thymine will pair with the adenine in DNA, but in mRNA it is reverse because thymine is absent in mRNA, messenger RNA. If we start with reading C, then the C will complementary in mRNA with G, guanine, and T will complementary with adenine, and adenine in mRNA complementary with the uracil. Okay, in this way, the DNA polymerase will started to read the entire the gene which it wanted to copy it. One thing here we have to notice that the head of the RNA polymerase, the DNA stripes are separated, where back of it is closing. So like this way the entire the DNA copy is read by the RNA polymerase. Once it is completed reading this one and preparing the mRNA strand, it separates from the DNA, and once this is done the DNA will enclosed and it we left with the mRNA, messenger RNA, which is ready to leave the nucleus and go ahead to the ribosome, where it can be read by the ribosome to prepare the nutrient protein. The piece of the information on mRNA is also called as template strand. Now this mRNA is totally ready to leave from the nucleus of the cell to the ribosomes which are there in the cytoplasm of the cell. And after this, it is very important to understand, like on DNA and on mRNA, the chemical bonds are bind together in a three bonds that is called as triplet or code. Triplet or code because three chemical bonds together form a code, and about 20 amino acids are required to form a new protein on mRNA. The mRNA will be read by the ribosome, and if we take this one, the CCA is for an example of proline amino acid, and AGU is an example for serine amino acid. You need not to remember these examples, it is just to illustrate. And after leaving this mRNA from the nucleus, it go and attach to the one of the ribosome which is in the cytoplasm. The ribosome and the mRNA together will bond uh, a bond, and the ribosome will start reading the information which is on the mRNA, and which gives the information to a transverse RNA to bring the amino acid with anticodon. You can see anticodon here on the tRNA with an amino acid, and this tRNA with anticodon will read the code on the mRNA and will go and attach to that mRNA. So A with U and G with C and U with A. This will go and attach with the mRNA. As the triplets chemical bond on tRNA, UCA will attract by the AGU on mRNA. This is the chemical code of the triplet which are paired together, which will attract the particular code of the information on tRNA, and it is for an example of the serine. This a code AGU is the code for the serine, and next code is the CCA, which will attract the amino acid of on tRNA GGU, which is a codes for proline, So likewise, and the ribosome will start move on ahead as it is reading and brings the correct amino acids in the correct order and joins the amino acids together to form a chain of amino acids. See, once it is finished reading of one bond and again it reads to the other chemical bond triplet form and brings the tRNA which is related to that same and the mRNA. So now what happens, tRNA molecule will fall down and leaving the amino acid back, and this process will continue throughout the mRNA strand is completely readed and coded by the mRNA amino acids. So once these amino acids are formed, okay, these ribosomes will deleted and the chain of amino acids formed together form a particular protein. This is all about this process. Thank you for watching. Please share and subscribe, and I'll come up with a new video. Thank you.